Hi, my name is Aiden and my biology role model is Dr. Bonnie Bassler, a molecular biologist. She's the chair of Princeton's Department of Molecular Biology and is currently studying cell to cell communication as well as the mechanisms they use to communicate through a process called quorum sensing, which we'll discuss later. Her hobbies include taking singing and acting lessons, aerobics class, hiking, bike riding, reading, cooking, and watching live theater. She was born in Chicago, Illinois in 1962, but spent her teenage years in Danville, California. She was always interested in critters and doing experiments, and one experiment she did when she was younger was in the back of a kid's magazine where she made a little paper steamboat powered by a candle. It was through experiments like this that she discovered she did not want to pursue physics or engineering. However, her interest in critters is how she decided she wanted to be a veterinarian. Her parents helped her explore this interest when she was 13 by helping her get a summer job as a veterinarian's assistant at the Miami Zoo and later at a local dog and cat clinic. She obtained her bachelor's in biochemistry at the University of California, Davis. It was during her time here that she participated in a research lab led by Rick Troy, who had two projects going on, studying cancer and studying bacteria. Dr. Bassler wanted to do cancer, she ended up in bacteria. And it was through this research lab that she discovered her love and fascination with bacteria. She later obtained her PhD in biochemistry from Johns Hopkins University and did her postdoctoral work in genetics at the Aguron Institute with Mike Silverman, where she was exposed to the concept that bacteria communicate with each other. During her junior year of college, her mother was sadly diagnosed with colon cancer and passed away 4 months later. It was due to this event that she was interested in Rick Troy's lab research in cancer and why she applied to join it. She also struggled, and still does to this day at points, to secure funding for her research projects as most people don't understand the importance of why we should further research on how bacteria communicate. There are many things I admire about her. One of the biggest things is her passion for making advancements in the medical field. Even though she decided not to go into veterinarian sciences, she went into a scientific field that allows her to make advancements in healthcare, and that's interesting to her. I also admire the humongous discovery she's made in the field of microbiology with cell communication. I love microbiology, so getting to learn about the ginormous discovery she made, it's awe-inspiring, truly. I also admire how, when faced with adversity, she never gives up. She's not held back, she doesn't stop or pause, she keeps challenging herself and moving forward. The article I'm going to discuss was published on July 31st of 2023. Natural silencing of quorum sensing activity protects Vibrio parahemolyticus from lysis by an autoinducer detecting phage. It looked at how a strain of Vibrio parahemolyticus, a virus that infects bacteria, had its mutations within it that prevent it from responding to autoinducers that its host bacteria receives. To understand this, we need to first discuss quorum sensing. Quorum sensing is, at its most basic sense, a way that microorganisms communicate with each other. They release a molecule called an autoinducer to sense the number of microorganisms around them, and when there are enough around them, they can change their gene expression and coordinate their behaviors with the microorganisms around them to better adapt to their environment and any changes that may occur. What they found was that Vibrio parahemolyticus is able to suppress their ability to receive autoinducers, preventing them from receiving information and halting its ability to reproduce effectively. Panel A of our figure shows fold changes of all quantified genes in Vibrio parahemolyticus as well as gene expression. The brighter the color, the more expressed the gene is. The panel to its right shows fold changes to a specific gene of Vibrio parahemolyticus. Panel B of our figure shows the output of PGP69-Lux, a bioluminescence signaling gene that allows researchers to track viruses. They put it in Vibrio parahemolyticus strain 882. An increase in output means there's an increase in gene expression within the virus. Panel C is relative to panel B in that it shows the different strains of bacteria strain 882 and whether it's carrying the virus or not. Dr. Bassler is currently working with her team to synthesize molecules that are similar to AL-2 autoinducers, which allow bacteria to carry out interspecies communication. The hope is that by further researching this autoinducer, we can develop new and more effective drugs that can handle antimicrobes. Thank you for listening.